IB Bio, Diversity, Evolution, Classification Part 5 has its focus on cladistics. Cladistics is the study of classification of organisms based on the branching of groups from a common ancestor. Cladistics is a system of classification based on the presumed phylogenetic relationships and the evolutionary history of groups of organisms. The essential idea is the ancestry of groups of species can be deduced by comparing their base or amino acid sequences. Here is the outline of the movies available for this unit. Use the outline to choose the movie you need. This movie has its focus right here. From here to the end of the movie, we will look at the study of evolutionary relationships with an eye on the information provided by cladograms. Before I present the first cladogram, I will provide you with a range of IB syllabus statements and related background. Phylogeny is the study of the evolutionary history, evolutionary origin of an organism or a group of related organisms. Here's a diagram displaying the evolutionary history of all organisms on Earth. The diagram displays the three domains, the archaeans, the eubacteria, and the eukarya. The common ancestor is here. Archaeans are prokaryotic cells that tend to live in extreme environments, very hot environments, very salty, etc. Archaeans have certain characteristics that suggest that they are more closely related to eukarya than eubacteria are to eukarya. Here are the first two IB syllabus statements of the movie. Define the term clade. State that cladograms are tree diagrams that show the most probable sequence of divergence in clades. A clade is a group of species that includes an ancestral species and all of its descendants. In other words, a common ancestor and all of its descendants. In this evolutionary tree drawn by Darwin, we could identify many clades, but let me give you one example or two. This is a clade. This is the ancestral species and all of its descendants. Here is a clade. This is the ancestral species and all of its descendants. Here's a chance to test your understanding of what constitutes a clade by examining the shaded regions of each evolutionary tree. The examples at the top are clades, as they include an ancestral species and all descendants, an ancestral species and all descendants. But the bottom examples are not clades, because the ancestral species of these groups here is not shaded, while in the tree on the right, a descendant group of this ancestor, this descendant group of this ancestor, is not shaded. Cladistics is a method of organizing information to reflect how organisms are related, the common ancestry of organisms. We use DNA base sequences, we use amino acid sequences in protein, and we use morphology, body form. This is the least important, although the most commonly used historical piece of evidence to produce cladograms. We can see a cladogram here. This diagram, this cladogram, is based on morphology, body form. An example of a clade within the cladogram would be here, an ancestral species with hair and all descendants. So let's analyze cladograms to deduce evolutionary relationships. Here we have a clade, an ancestral species, and all descendants. This is an ancestral lineage, an ancestral species with a branch point representing a divergence of two groups, which is the first step in the formation of new species, called a speciation event. Let's focus on two species, B and C. Here is their shared history. Here is the unique history of B, and here the unique history of C. The common ancestor of B and C is right here. Here is the common ancestor of A, B, and C. Remember, the strength of cladistics is that the comparisons are objective, relying on morphological and molecular homologies. The weakness of cladistics is that molecular differences are analyzed on the basis of probabilities. Improbable events occasionally occur, occasionally making the analysis wrong. In the last slide, morphology, or body form, was used to build a cladogram. Cladograms attempt to elucidate the evolutionary history of organisms, but scientists must be careful with the use of morphology, or body form, because sometimes we can interpret structures that appear similar to have a common ancestry. For example, are the wing of a bird and the wing of a butterfly of common ancestry? No, they are analogous. Homologous structures, often structures with different functions, have common ancestry, but analogous structures do not. Cladistics is meant to understand the true evolutionary history of species, species that have common ancestry. DNA base sequences or amino acid sequence are much better evidence of common ancestry than the use of morphological characters. Thus, that brings me to the next IB syllabus statement. Describe the primary evidence used to determine evolutionary relationships, clades. The primary evidence for cladistics would be a comparison of DNA base sequences or a comparison of amino acid sequences in polypeptides. You will see morphology used in cladistics, but it is by far the least reliable evidence. The last slide on analogous structures provides the perfect example of why body form is not reliable evidence of evolutionary relationships. The data on this slide represents the number of base differences between different species for a single gene known as cytochrome C. You can see that the number of base differences between the human and the monkey is 1, while the number of base differences between the human and the fly is 33. The number of base differences is proportional to the time that has passed since the divergence of the two groups from the common ancestor. For example, the common ancestor of the human and the monkey lived relatively recently. Thus, there are few base differences in this cytochrome C gene. However, the common ancestor of the human and the fly existed quite far back in time. From the point at which populations split and evolve in separate directions, mutations occur, selection occurs, and changes in the genetics of one population are no longer shared with the other population. This is known as reproductive isolation. The number of base differences, mutations in the DNA of each species, is proportional to the time that has passed since the split, since the divergence from the common ancestor. Thus, we can build a cladogram knowing that the human and the monkey are more closely related than the human and the fly because their common ancestor lived more recently in time, thus less time has passed for mutations to accrue. So that takes us directly to this IB syllabus statement. Explain the positive correlation between the number of base or amino acid differences between two species and the time since they diverged from a common ancestor. Here's a cladogram based on DNA nucleotide base differences in the cytochrome C gene. Two species, such as human and snake, share a common ancestor here. Humans and pigs have a common ancestor here. As a result of the fact that the common ancestor of the human and snake lived further back in time than the common ancestor of human and pig, there's been more time for mutations to accumulate, resulting in a larger number of differences in DNA base sequences between the human and the snake at 20 relative to the human and the pig at 13. Mutations accumulate at roughly a constant and predictable rate. Therefore, the number of differences can be used as a molecular clock to measure the time since two divergent groups shared a common ancestor. For example, if the number of base differences between two species is 50 mutations, then the date of divergence is approximately 90 million years ago. If the number of differences in the DNA base sequence between two species is lower, let's say 15, then the date of divergence is approximately 25 million years ago. In other words, the common ancestor of the two species being compared lived 25 million years ago. To construct a cladogram, as you can see here, one would use DNA base sequence or amino acid sequence data. The timeline runs along the x-axis from past to present. The more differences in the base sequences of two species, A and F by way of example, the further back in time the common ancestor. Molecular clocks or fossil record data can be used to provide the age of the common ancestor. Fewer differences in, say, amino acid sequences of a specific protein, species A and C, by way of example, would indicate a relatively recently lived common ancestor. Molecular clocks using a known mutation rate or fossil record data could provide an age of the common ancestor. Here is a cladogram based on body form, morphology. Remember, be somewhat skeptical of cladograms based on body form as compared to cladograms based on DNA base differences or amino acid differences in polypeptides. Based on what we have studied already, you should recognize the following animal phyla in this diagram: Porifera, Cnidaria, Platyhelminthes, Arthropoda, Annelida, Mollusca, and Chordata. Study this slide to refresh your understanding of the differences among these groups. Here is a cladogram displaying the evolutionary relationships within the plant kingdom. You should recognize the terms that distinguish the bryophytes from the phyllocynophytes, from the coniferophytes, and the angiospermophytes. This slide provides a cladogram displaying the evolutionary relationship among hemoglobin molecules, here, based on the number of amino acid differences. A detailed analysis of the hemoglobin family of molecules suggests that myoglobin was the ancestral molecule. At this point, you should be able to handle the following IB syllabus statements. Analyze cladograms to deduce evolutionary relationships. Analyze cladograms that include humans and other primates. So here is a cladogram that includes the humans and other groups of primates. Here is a clade that includes the ancestor of humans and apes and all descendants. The ancestor of apes and old world monkeys is here, and the clade would be defined by this circle. You should be able to see that humans and chimpanzees are more closely related than humans are to orangutans, because the ancestor of humans and chimps lived more recently in time relative to the ancestor of humans and orangutans. There would be fewer base differences for most any gene between the human and the chimp, with a common ancestor having lived more recently, as compared to the human and the orang, whose common ancestor lived further back in time. So here's a question. Which two species are more closely related, the human and the gibbon, or the orangutan and the gibbon? Now you have to be careful. The human and the gibbon have a common ancestor here, while the orang and the gibbon have a common ancestor here, the same common ancestor. In other words, just because the orang and the gibbon sit side by side on a cladogram, you must look back to their ancestor and take note that the orang and the gibbon are just as distantly related as the human is to the gibbon. Here is a phylogenetic tree for humans and closely related apes. Humans and chimps have a common ancestor here. Humans and gorilla have a common ancestor here. Chimps and gorilla share the same common ancestor that humans share with gorilla. Humans, chimps, and gorillas are all equally related to the orangutan. The common ancestor of all four groups is here. This cladogram nicely adds time so that we can see the time of divergence for the various groups. The common ancestor of humans and chimps was approximately 6 million years ago, but the divergence of apes from old world monkeys occurred more than 20 million years ago. Hopefully this IB statement should be easy. Explain the positive correlation between the number of differences, base or amino acid differences, between two species and the time since they diverged from a common ancestor. For example, we have base sequences for two individuals of the same population. That's before diverging. Then they diverge, and we get genetic change in each separately, and the genetic changes are not shared. That's reproductive isolation. Certain segments of the DNA remain homologous, providing evidence of their common ancestry. An analysis of the base differences, along with known rates of mutation, would provide the molecular clock to determine the age of the common ancestor. Here you can see the genetics of the two divergent populations. The homologous sequences are in yellow and orange. Computers do the work of determining the genetic differences among species, and then using known rates of mutation, which is the molecular clock, the age of the common ancestor can be determined. Computer analysis indicates that the base differences in a certain gene to be 40% between human and a plant, but only 30% between human and the fungus. The human and the fungus have a more recently lived common ancestor. The fewer number of base differences indicates less time has passed since their common ancestor. Thus, the human and the fungus are more closely related than the human is to the plant. Just as a note, the ancestry of the fungus to the plant is equivalent to the ancestry of the human with the plant. So here is the cladogram that would be constructed based on the data from the last slide. You can see that humans are more closely related to mushrooms with their common ancestor here than humans are to plants with the common ancestor here. Since the base difference between the fungus and the plant with the common ancestor here is approximately the same as the base difference between the human and the plant with a common ancestor also here, you can see that humans and mushrooms are equally distant from the divergence with plants. This slide gives us an index of base changes among HIV sequences through time. Assuming constant rates of mutation, an analysis of base differences among the various strains of HIV allows a regression line to be drawn that would provide evidence of when the common ancestor of all HIV strains existed, approximately 1930. Let's continue with an analysis of cladograms to deduce evolutionary relationships. Here is a phylogenetic tree displaying the evolutionary relationships among species. Historically, phylogenetic trees were built using body form as the only evidence. Today, cladograms are more accurately constructed using DNA and amino acid sequence data as evidence. I've circled a clade. Review your understanding of a clade. It is an ancestral species and all of its descendants. Could you speak specifically about evolutionary relatedness using this diagram? Compare the wolf and the domestic dog as compared to the wolf and the otter. Speculate about the degree of DNA base differences in the genes of these three species. One last note. The use of the 7 taxonomic categories for classification is very arbitrary. There are no guidelines on how a species is classified other than at the species level. Why are these two species put into a different genus rather than the same genus? The 7 taxa hierarchy, kingdom, phylum, class, order, family, genus, species, can be helpful or confusing. When cladograms are constructed using DNA base sequence data or amino acid sequence data, we know the common ancestor of the wolf and the coyote lived more recently than the common ancestor of the wolf and the otter. Since the common ancestor of the otter, the coyote, and wolf is here, we would predict similar numbers of base differences between the otter and the coyote as compared to the otter and the wolf. While this cladogram does provide information on relatedness, we don't know anything about time, the time at which common ancestors lived or groups diverged, unless we were given more information. And at the top, you can see that we're given the classification hierarchy. The taxonomic hierarchy are not as well defined as this diagram would make you believe. For example, why the badger and the otter are placed into separate genera is not clear, nor why are the otter and the badger placed into a separate family from the canines. And remember, phylogenetic trees using body form data, morphological data, will be less accurate than cladograms using DNA base sequence data or amino acid sequence data. Now here's some data for various groups of organisms using morphological information. Could you build a cladogram with this data? I'll let you stop the movie now to give it a try. Here's the cladogram. How did you do? This cladogram assumes equal rates of mutation in each line from the common ancestor, the point of divergence. Is this assumption correct? Phylogenetic trees can provide time, as you can see here, but remember, how would time come to be known? Radioactive isotopes in the rock within which fossils are found, or known mutation rates in combination with base differences for a gene, the molecular clock. This cladogram shows the evolutionary relationships among the great apes, including the orangs, the chimpanzees, the humans, and the gorillas. You can see the arbitrary nature of the 7 taxa hierarchy right here. All 5 of these species have been placed into the same family, Hominidae, but the orangs are clearly more distantly related. And by what criteria were the chimpanzees placed into their own genus, the pangenus, when they differ from humans by only about 2% of their DNA? How much genetic difference would specify a different genus or a different family? With only a 2% genetic difference between humans and chimps, couldn't chimps be placed into the Homo genus alongside humans? The figwort family had been classified based on morphological data, but more recently, DNA evidence has resulted in new relationships. Some former figworts are now placed in different families. And that brings us to the end of IB Bio Diversity, Evolution, and Classification, Part 5.